Wanted to thank everybody for coming out today. Today I'm going to be going over my master's proposal, which takes a biologically informed approach to natural product drug discovery using a zygomycete fungus, Basidiobolus meristosporus. I'm going to start first with some background information about the biological system I'm using and the rationale for choosing this system. And then I'll go into my hypotheses that I've generated, the research design for testing those hypotheses, and then a little bit of a discussion about my preliminary findings and an outline for the rest of my master's project. So the system that I have chosen to work with is a strain of Basidiobolus meristosporus isolated from gecko dung. Members of this genus live as gastrointestinal symbionts of amphibians and reptiles. One particularly interesting feature of this species that plays a role in making it a good candidate for natural product discovery is that it has high levels of horizontal gene transfer from bacteria, which I will come back to later. But to give you a little bit of an idea of what this organism looks like, I have a couple pictures up here, some of the different morphologies. So typically in culture, you see septate hyphae as the main growth form. And then over here on the left, you can also see this really interesting cell type called the palmella state, which is this interesting binucleus state that isn't often observed. And then we have one of the asexual spore types on the right here, which is a ballistoconidia, which is used to forcibly discharge these spores into the environment. A little bit of background of fungi as a kingdom in natural product discovery. Typically, the phyla that are used for natural product discovery come from the Basidiomycota, so the fleshy fungi, the mushrooms and the conchs, and then also the Ascomycota, the sac fungi like Penicillium, zygomycetes, which is an informal grouping made up of two phyla, Mucoromycota and Zygomycota, are currently the focus of this interinstitutional project called ZygoLife, which my project is a part of, and it's focused mainly on furthering our understanding of the evolutionary history and the ecological associations of these understudied fungi. Basidiobolus is rather unique, even for a zygomycete in a few regards. Firstly, its phylogenetic placement isn't resolved. It's typically considered to be part of the insect destroyer, so the Entomoptera mycotina. And furthermore, while zygomycetes are typically considered to be poor producers of secondary metabolites, initial analyses of Basidiobolus have shown that it's an exception to this trend. In addition, it also has a very complex life history. So as I mentioned previously, most of this genus are amphibian and reptile endosymbionts in the gut. And they typically dwell as endospores or as this gut stage type cell rather than as hyphae. And because they're in this close interaction with all of these microbes here, that's one of the indicators that there might be some sort of antimicrobial capabilities of the species. So eventually some cells are excreted into the dung where those ballistoconidia I showed you can form and discharge into the environment, where they can go through asexual or sexual repeating cycles out in the environment. Eventually, one of the asexual spores will adhere to some sort of insect, and then through a spore-bearing insect, it will eventually make its way to colonize the gut of a host. Next, I wanted to talk a little bit about secondary metabolites, particularly from fungi, but before I get into that, I do want to clarify this terminology of secondary metabolite and natural product. So secondary metabolites are any organic compounds produced by an organism that aren't used in basic processes like growth, development, and reproduction, whereas a natural product is just any compound that is produced by an organism in nature. So while there are some distinctions between those terms, I'm going to be using them interchangeably, as all of the natural products of interest in this natural product discovery are secondary metabolites. So there are these different classes of secondary metabolites, but 4 of them are common secondary metabolites produced by fungi, and these include peptides, polyketides, terpenoids, and alkaloids. So each of these secondary metabolites can show a wide variety of functions, and to go over some of these different classes and functions, I have some examples here. So terpenoids, which as the name suggests are terpene derived. A good example of this is gibberellic acid, which most people are familiar with as a plant signaling hormone, but it's also produced by some fungi like the rice pathogen Gibberella fujikori, which is actually the namesake for the compound. And then there are alkaloids, which are nitrogenous compounds, typically having some pronounced physiological effect on humans. One fungal example would be ibotenic acid, which is the main psychoactive component of Amanita muscaria. And then there are these polyketides, which are typically derived from acetone. So they have these alternating carbon bound to oxygen with these methylene, these CH2 groups in between. And one example of this would be fumonisine, which is a toxin produced by Fusarium. And then what I will be focusing on from here on out for the most part are these non-ribosomal peptides, which is a class of secondary metabolites that has these amino acid backgrounds. So in this circle here, you can see all the amino acids that make up this backbone for the compound. And one example that I'm going to be focusing on is cyclosporin, as it comes from a fungus Talipocladium. And it's widely used in medicine for its immunosuppressive activity, such as in organ transplants, where it helps the recipient accept a donated organ instead of rejecting it as a foreign invader. So, as I mentioned previously, Basidiobolus has a lot of horizontal gene transfer from bacteria, and this is one of the ways that we can biologically inform what makes a good candidate. So approximately 3 to 5% of all of the predicted gene models for this species are a product of horizontal gene transfer. And as you can see, there's a diversity of taxonomic groups that contribute to these horizontally transferred genes. And most of those taxa are associated with amphibian and GI tracts. So it's very likely that this intimate interaction where genes can be transferred is happening in that in the GI tract of the host. And while you can't see in this figure, other analyses of these genes have shown that approximately 50% of all of these horizontal genes encode for some sort of secondary metabolite product, leading to the conclusion that there's some sort of selection pressure for Basidiobolus to be producing all these secondary metabolites. Another route for using genomics to biologically inform this is to look at the core genes that are involved with SM production. So you can see here that Basidiobolus has a large abundance of these core genes involved in secondary metabolite production that compared to its other zygomycete relatives. So of these core genes, there's these polyketides synthases. So the PKS and PKS-like, there were 4 genes that were predicted, 10 for those terpene cyclases, and then the overwhelming majority of them appear to be these NRPS, NRPS-like. So there's like 30 of those. These core genes are often part of biosynthetic gene clusters, and these core genes are often modular in nature. So a good example of this is the biosynthetic gene cluster with the NRPS core gene that it encodes for the protein products needed to synthesize cyclosporin A. So if you look at this biosynthetic gene model at the top here, the SIM-A gene is the core gene for producing the SIM-A NRPS, and all of these other genes are involved in things like transporting the intermediates, outpumping of the final product, modifying the SM products, along with many other functions that are needed to produce the secondary metabolites. In the second figure down here, you can see the SIM-A enzyme, which is a massive protein comprised of over 15,000 amino acid residues. So this protein is comprised of these different modules, and each of these modules has at least 3 domains, which are the adenylation domain, the thiolation domain, and the condensation domain. And together, these domains are essentially responsible for selecting certain amino acids and then adding them to the growing amino acid chain. In this example, as there are 11 of these modules, you have an 11 amino acid backbone that then can be cyclized and turned into the final cyclosporin product. So these different domains, the adenylation domain is responsible for recognizing and activating specific amino acids. The thiolation domain covalently then links that amino acid to the enzyme, and then the condensation domain links the amino acids together to grow the amino acid chain. Because these A domains are necessary for identifying and activating specific amino acids, and because they are conserved structures, we can use these to construct phylogenies through a protein analysis. So the A domain analyses indicate that Basidiobolus meristosporus contains these NRPS A domains to produce products from many different clades. So one of these clades is the cyclosporin-like clade, which is involved in producing not just cyclosporin, but other depsipeptides and other species of fungi, particularly ascomycetes involved in as insect pathogens, use these in interactions with their animal hosts. So given that Basidiobolus interacts with an amphibian or a reptile host, this has led me to hypothesize that such a compound from this clade would possibly show some sort of immunosuppression as an activity. Additionally, there are these other compounds that are likely involved with antimicrobial activities. So there are these siderophores, which are important in iron sequestration. The GI tract is an iron-limited environment, so compounds produced in the siderophore clade likely function in microbial competition. And then there are compounds like the surfactant-like molecules, which have been shown to promote and inhibit growth of certain microbes. Surfactants act by embedding themselves in and disrupting membranes. As such, the products in these clades would likely be important in direct interactions with other microbes. So all of these different genomic and proteomic analyses have led to three hypotheses. So the first is that Basidiobolus will be producing biologically active small molecules. And this might seem kind of obvious, but we don't have any descriptions of compounds. And so far, all that data that I presented comes from predictions of genomic and proteomic analyses. So this is really the first time that people are looking for the actual production of these compounds that have predicted gene models. And then the second and third are a little bit more specific and build on that extracts of these small molecules will display an immunosuppressive activity and will display an antimicrobial activity. So to test this, I'm going to be growing Basidiobolus in 5 different media types to increase the profile of secondary metabolites being tested. Compounds will then be extracted from the mycelial tissue, and these crude extracts will be used for bioassays on several cell types, including HeLa cells, which are a human cell line to look at sort of a general cytotoxicity, raw macrophage cells to look at immune modulation or immunosuppression, and again, some World Health Organization priority 1 fungal and bacterial pathogens to look at different antimicrobial activities. So then positive hits from those initial bioassays will be further fractioned using liquid chromatography to increase the purity of each fraction. And then the bioassays will be rerun with those fractions to narrow the range of compounds that are in these fractions that are producing some observable activity. Eventually, these semi-pure fractions will be characterizing using mass spec to look at the profiles of the active fractions. In order to validate this differential production in the different media types, mass spec data of crude extracts is going to be used to determine the metabolite profiles for each media type. And RNA is going to be extracted from mycelial tissue, and RNA-seq will be correlated with those mass spec profiles to validate that Basidiobolus is responsible for production of these compounds, but also to look at some sort of genotype-phenotype linkage with the profiles from the active fractions. Moving on to the preliminary findings that I have so far, analysis of these crude extracts has confirmed that Basidiobolus is showing differential production of these metabolites based on media type, and that some of the observed compounds that we're seeing are truly, in fact, peptidic in nature. The PCA on the top left shows these profiles of each media type based on all of the compounds detected via mass spec. The media types that are closer together show more commonalities in their profiles, and ones that are further apart are more different. Additionally, there are these individual points inside of each of these circles that group together that show that there is a consistent correlation between the media type and the observed SM profile. And then I have a heat map on the right there that also indicates these similarities and differences, where the darkness of each band indicates the observed intensity of a compound, and then bands that align vertically indicate the presence of that compound either between media types or between replicates of a given media. So for example, we have a compound like this one here that is shown to be produced in abundance in just the cornmeal extracts, but is not abundant in any other media type. And then we have bands here which show that we have compounds that are produced in 3 out of the 5 different media that we're looking at. So we have compounds that are produced by just one media or are sometimes present in more than one, but there is this differential production based on the media type. And then this graphic at the bottom here shows the MS-MS spectrum for just a single compound. Along that x-axis, you have a display of a mass to charge ratio, and each of the peaks, the height of the peak represents the intensity for an ion at that mass to charge ratio. So the difference in these peaks is useful for investigating the fragmentation patterns of a compound, and then the difference in those peaks can be used to identify residues of that fragment. So for this particular example, there are fragments that show that there are peptid or amino acid residues, showing that we have compounds that are peptidic in nature, which is expected based on that abundance of NRPS genes that we saw from the genomic predictions. The other preliminary data that I have so far is an initial bioassay against a gram-positive and a gram-negative bacterium. So this red line here that you're seeing across the screen marks a 50% cell survival, and this is a metric used as an early indicator for a positive hit to further explore. So it shows that the extract is displaying some sort of significant activity. So in our gram-positive test, all 3 of the extracts from this cornmeal media type and 2 of the 3 from the yeast malt showed this significant activity, whereas none of the extracts tested showed any significant activity against the gram-negative bacteria that we tested. So this shows that there might be some sort of specificity, and whether or not that's between gram-positive and gram-negative, there's at least some sort of specificity beyond just a general cytotoxicity from these extracts here. To go through my future work, by the end of summer, when we have things back up and running, I would like to have rerun some of the mass spec on the crude extracts to validate the PCA that I showed you and to add more data to increase the resolution on the PCA. And then also run those first crude extracts on those cell types that I mentioned, the HeLa cells, the macrophages, and the priority pathogens. And then to perform the first fractionation of positive hits to get ready for the second round of assays, which then by the end of fall, I'll have rerun those assays with those more pure fractions and use the mass spec to characterize those semi-pure fractions, leaving me to, in winter, be able to extract RNA from the mycelium that I've saved and to do that correlation analysis to begin generating hypotheses about the genotype-phenotype linkage, and to validate that the compounds from the active fractions are being produced by the organism that I'm working with. So wanted to say thank you to all of my committee members for your help and your feedback so far. Thank you to the Spatafora Lab and McPhail Lab, and to my cohort for all of the support that I've gotten so far. And then a special shout out to Javier Tabima and Richard Tijon for helping me develop all of my laboratory and analytical techniques as an undergrad with y'all and up until now. So yeah, thank you. Do I have any questions?
All right, the floor is open for questions. You can put them in chat.
Do you mind if I?
Go ahead.
Again, this is Tom. Yeah. Hey, given the conditionality of these different media for metabolite expression, I'm just curious: is there any kind of media that might replicate, you know, more of the natural habitat of this bug? Something like, you know, intestinal fluids, or I don't know, serum, something like that, where you might look for perhaps more biologically relevant. I mean, I'm not saying they're not all biologically relevant, but you know what I mean? Some kind of ecosystem that it's more attuned to. Does that make sense?
Yes, that makes perfect sense. And that's actually
by the
something
way, nice talk too. Thanks. Yeah.
Thank you. Yeah, no, that's something we've discussed kind of at length. And so one of the issues there, and I'm going to back up here to talk about part of this. So one of the issues there is that there doesn't appear to be in my research a good amphibian gut type media. So, you know, maybe you could do something intestinal, looking at the human side of it or something like that. But there doesn't seem to be a good media type to work as a true proxy for the host that you would see for Basidiobolus. Most of the work that's done with amphibians and the microbiomes looks at, you know, like skin microbiomes and things like that. So there's not really a good media to use there. But fortunately, so looking at this A-domain phylogeny, you know, you're right, we might be missing certain compounds that might only be expressed in a true like gut condition. But this phylogeny showing different compounds that are being produced by our organism were taken from, you know, like the genome that was created from the culture, and they used I want to say they used like a yeast malt type broth as well. So there definitely is some sort of activity that we're going to be able to see from these media types, and we do get you know, compounds that it would be producing in nature. But specifically looking at that gut, yeah, that's something that we don't really have a media type for right now.
Thanks, appreciate it.
Other questions for Ian? Don't be shy. All right, well, not hearing any. Let's all unmute and give Ian a big thanks.